there. Sorry about that, Darius. All right. Now they can hear me. They missed the part about the pumpkins. Our main focus right now is the R1, and that is focused on the diabetes and glycation. But we have been pursuing funding. We have one active R1, another one scored, and we have another one we're working on in this area that we got recently scored and we'll be putting back in, and then some of the protein nutrition work. So I'm going to spend most of my time on this, but touch on these a little bit to give you a sense of what we're doing. Okay, that figure out it. So since everybody doesn't do tendon here, I'd like to give you a semester crash course in tendon physiology in 3 slides. So the tendon, I really got fascinated with it during my postdoc. And just because, it was at the time, which was 2004, it was very, very understudied. And we really didn't know much about it, but it's one of the most commonly injured tissues in the body. And so it just was a really interesting area to me. And to kind of set it up here, the tendon, obviously we all know it connects muscle to bone, but the tendon is contiguous with the connective tissue layers of skeletal muscle. So if you look on the right side here, this is from Michael Kier's group in Copenhagen. This is a cross section of skeletal muscle where they've actually digested out the muscle, leaving just the connective tissue web. This is where a single fiber would be. And so when we initiate cross-bridge cycling and generate tension, that tension has to go somewhere or the muscle would be functionally useless. And that tension developed in our cross bridges gets transmitted into this web of connection tissue that goes through all these layers, ultimately to the fascia around the muscle, the tendon, allowing the bone to move. So if there's anything wrong with that network, it's a problem. To give you a little bit more about how the tendon is organized, this looks a lot like your muscle physiology picture, where we go from the whole muscle down to the muscle fiber. Now, the tendon doesn't have sarcomeres and stuff like that, but when we look at the tendon, we study it at different levels because different things can happen at different levels. I'll show you some work on whole tendons, but we also break it down into some of these smaller parts down to the macro scale and nanoscale level, where we're measuring collagen, which is the main structural component of tendon. Tendon is about, collagen is about 50–55% of the dry weight of tendon. So it's a very large amount of one single protein in a tissue. And we do studies on the tendon fibroblasts, which are the primary cell type in tendons that control and remodel the tendon as well. And what's really neat is if you look even closer down at the smallest levels of the tendon, then if you look at the amino acid sequence of collagen, it's this really unique kind of repeating sequence of glycine, proline, hydroxyproline with a few other amino acids randomly thrown in there. And the collagen actually forms this triple helix structure that you see here, and ultimately gets set up in this format here that you see with these various collagen helices. And these are then normally cross-linked, enzymatically cross-linked to support the structure. Now we're going to talk about non-enzymatic cross-linking and how that works later on. So that's the basics of the general structure of tendon. Now, why is the tendon important? Okay, from a functional standpoint, when most of us think of tendon injuries, we often think of traumatic ruptures. If you've ever heard like the Achilles, if it ruptures, it sounds like a gunshot, and it's very traumatic. The rotator cuff is a common site, your elbow tendinopathy. But most tendon problems are actually non-traumatic injuries. It's tendon pain, degeneration, things like that cause most of the clinical problems, and that can be very chronic and be very difficult to treat and deal with. So why is that important? Well, we're going to use this analogy of a bicycle to describe how the tendon works. This is one of my favorite figures. And if we look at this bike analogy here, we have our bone, which is the wheel. We have our tendon, which is the chain connecting the wheel to the muscle, which is our pedals. And that pedals are connected to the chain through the intermuscular connective tissue. Okay, so when you're riding your bike, you're applying force to that intramuscular connective tissue that then pulls on the tendon or the chain, turning the wheel or the bone. Now, just like a bicycle chain, if you have a rusty bike chain or a loose bike chain, you're not going to be as effectively be able to move that muscular force into movement. So just simple changes in the structure of that chain, or in this case of the tendon, can have consequences for the function of the muscle. If you could have a perfect muscle that's perfectly intact with no issues, but you have a damaged tendon, then that force isn't going to be effectively transmitted, and you're going to see reductions in things like muscle performance, muscle power output, regardless of the muscle itself. So that's why there's multiple reasons why the tendon is important. It ruptures, it can have chronic pain and degeneration, or just changes in the properties of the tendon can change function. So this is where we move into our actual research, and I'm going to tell you a little bit about why we got interested in diabetes and tendons. Well, first, whoops, there we go. First, we know that patients with diabetes have a much higher incidence of tendon abnormalities and injuries. I was telling some of the faculty today, if you were to do an MRI or an ultrasound of 100 patients with diabetes, or approximately 90% of them would have some underlying tendon pathology. They may be asymptomatic, but it's very, very high rates. And hospital rates after injury are much higher than persons without diabetes. And this even moves into individuals with metabolic syndrome as well. So if they do injure themselves, they're much more likely to need hospital care. And on top of that, obesity, which is commonly associated with type 2 diabetes, is a separate risk factor for tendon complications. And another issue with diabetes is that the ability of the tendon to heal is reduced compared to a normal glycemic person without diabetes. And then when they do have to have surgery, they often have more complications than those without diabetes. So we have lots of different things going on. And most critically, and what led us to what I'm going to talk about, is there's quite a few studies now, both in humans and animal models, as well as dogs. I guess it's an animal model. Humans grown as a dog, that reaching clinical treatment goals like reducing A1C and things like that doesn't seem to prevent these tendon injuries, suggesting that hyperglycemia is maybe not the sole driver of these complications. And that's what led us to this research. And I'm going to explain how we got to that point and then show you some of our data. For many, many years, and still in many circles, the thought has always been that the problems in tendon with diabetes is something called non-enzymatic cross-link formation in your tendon. These what's called AGE cross-links. The AGE stands for advanced glycation end products. And I'm not a biochemist, so I'm not going to dig through every aspect of this reaction process. But basically, the nuts and bolts of it is that chronic exposure of proteins to glucose in the body through hyperglycemia, eventually those glucose molecules start to react to those proteins, go through a series of reactions that happens over hours, days, and years, ultimately leading to the formation of these AGEs that can then form cross-links with your collagen molecules, in addition to the enzymatic cross-linking we already have. Well, now you've added more structure, more of these things holding the tendon together, and that can make your tissue stiffer. And so for the longest time, that's what we thought was the problem. And some of that developed from a lot of ex vivo type studies that were really designed to strongly induce cross-linking using things like ribose or fructose. And so they kind of gave us this impression that this is what happens. But more recently, we're finding that maybe this doesn't actually happen to the extent that we thought it did. And I'll show you some of our data as well, but to give you some idea here, this is a Achilles tendon isolated from patients that were having their limbs salvaged due to chronic diabetes. And they basically took those Achilles tendons and put them in a device and pulled them till they snapped. and you can measure the failure load, stiffness, and some other properties. And what they found was opposite of what we would think. The healthy controls are on the right, and the patients with diabetes are on the left here. And all of our variables, our modulus, our stiffness, they're all lower rather than being higher in the patients with diabetes. So exactly the opposite of what we thought. And we're seeing similar things in DB mice, which is a mouse model of the type 2 diabetes and obesity. So it's not just in this single human study. So that was, okay, what's going on? If we're forming these AGE crosslinks, which we probably are, how come the tissue is not getting stiffer? Well, if we look more closely at biopsy samples from individuals with diabetes, what we see is a deterioration of the tendon structure. And this can just be seen briefly here. This is a normal looking tendon. You can see all the fibrils are aligned nice and straight. Now you look at individuals with type 2 diabetes, you can see it's kind of wavy and distorted. It doesn't have that perfect, nice alignment. Here you can see some gaps between the spacing, which is not good as well. So what that's telling us is that the tendon in these patients is more disorganized. So now it's more difficult for them to properly transfer that force to the muscle. And another group out of WashU, Jen Zellers, they followed up on some of this work, and they actually got Achilles tendon samples from patients, did some measurements to assess how aligned the collagen was, which is these two measurements here. And they also measured the AGE content in the tissues. And then they correlated that with our tendon stiffness or modulus to see, okay, if we have more AGEs, is the tendon going to be stiffer? Is basically the question they were asked. Well, if you look over here, you can see that there's no relationship between the AGE content in the tendon and the stiffness of the tendon, whereas the stiffness of the tendon was much more related to these markers of disorganization. So concluding that even if there is more crosslinks in the tendon, the degeneration and disorganization of the structure is overriding that and reducing the mechanical function of the tendon. So that led us to like, okay, well, what's causing this degeneration? Okay, what else could be going on? Well, there's another thing that these AGEs do. Some of them, they do not all form crosslinks in the tissue. Some of them basically end up in our blood as adducts, AGE adducts. So they end up in our bloodstream, and I'll show you some other sources that they come from as well. And lo and behold, we have this receptor on pretty much all of our cells, as far as I know, called the receptor for advanced glycation end products, or RAGE. Okay, and it's one of those receptors that we really not 100% sure all what it does. It has multiple ligands, but when it's overly stimulated, it leads to a lot of bad things in our cells: inflammation, oxidative stress, etc. But we didn't really know if it had any involvement in tendon at all. And what's more important was, as we knew from the literature, was that if we look at the blood from a patient with diabetes, they have a much higher AGE load. Their blood has much higher levels of AGEs. So we thought, okay, maybe these might be driving some of this process. We also, surprisingly, there's data now to suggest, we have some too, that these serum AGE levels are not related to blood glucose control. So even if you reduce your A1C, you still have high AGEs. So simply, hey, go make your blood glucose better is not going to solve the problem. So that led us to some of our first experiments and really following this hypothetical model. If the AGEs are elevated, does it impair tenocyte function through RAGE-mediated mechanisms? And does that lead to loss of function and such? And so that's what we sought to prove. And we gathered some of this preliminary data. I'll show you is what we used to get our R1 funded, and so we can pursue that question even further. So basically, this is the question that we wanted to ask: is, do these serum AGEs, could they contribute to tendon pathologies in patients with diabetes? So we started, and what I probably should have put these slides a couple slides earlier. I probably did it one time and I moved them around, not thinking about it, but The question I get asked is back to that, hey, if you're saying glucose is what drives AGEs, how come controlling my blood glucose doesn't solve the problem? Well, the issue is we get these AGEs not just from having hyperglycemia, but we get them from other exogenous and endogenous sources. And I'm going to show you a couple examples of those. One of these exogenous sources is a nutritional aspect, is what we eat. Okay, a Western diet is high in AGEs. Okay, the processed foods when we eat processed foods, it makes AGEs. They actually call it the Browning reaction. Okay, that nice yummy crust on your bread has AGEs in it. Your coffee has AGEs in it. Now, I will say we don't know with 100% certainty how well we know they're absorbed in the GI, but we don't know there's too many details about it yet. But if you eat a high AGE diet, your serum AGEs will go up. And that nice, you go have your nice barbecue. What are they doing? They're coating your meat with sugar and burning the sugar on there. Okay, yummy. It tastes great, but it's not so good for you. And it's not even just the content of the diet, but how you cook your food. So pan frying your foods increases your AGE content. And unfortunately, that yummy bacon that we all love is loaded with AGEs. Tip, microwave it, don't fry it, microwave it, and it has way less AGEs. So now where another issue where they come in, and this is really pertinent to patients with diabetes, is we do get them from endogenous sources. And the most common source is through glycolysis. So as you're pushing glucose through glycolysis, you generate methylglyoxal, which can then form MGH1, which is a potent AGE. Now, normally in a healthy adult, we have buffering systems to deal with this. We clear it out through what's called the glyoxylase system or GLOW enzymes. And they detoxify these AGEs, preventing them from building up. However, a friend of mine, Jay Koss, is a collaborator of mine, has shown, this is lean, healthy controls in patients with type 2 diabetes, that the GLOW enzymes that are in skeletal muscle buffer these AGEs are much lower. So individual with diabetes now they just don't have those buffering capacities. Not only are they getting them from glycolysis, but they're eating them in excess as well. So our first experiment that we did to prove kind of a proof of concept experiment was our very first cell culture experiment that I ever did in my career. I had a great PhD student who's now actually in a couple of years will make about 5 times as much money as me as an orthopedic surgeon, but that's okay. We're good friends. He designed this experiment, and he wanted to show that these AGEs do indeed do anything to tendon cells. So he set up an experiment where he isolated rat tenocytes from the Achilles tendon, and he treated 2 different glucose conditions, a kind of a classic low or normal and high experiment. And then he went in the literature and estimated what serum AGE levels would be in the patients and came up with a dose regimen for this study. And then he ended up making his own AGEs. You can do this in the lab by basically baking albumin with this reaction that drives the process. And basically what you end up with is the AGE-CML. We just call it AGE-DSA. And no, this is not the tap water out of my lab, even though it does look like that occasionally. This is remember I said that Browning reaction, this is our AGE solution. So imagine someone's blood with a lot of AGEs floating around, theoretically it might look something like that. And this is just the data. If we did not properly run the reaction, we would have free amine terminals, and we just demonstrate for proof of concept that we did indeed have what we wanted. So what we've done, we did several experiments, and I'm going to show you all the data because we would be here for another 20 minutes. But we did 2 different things, or 3 or 4 different things. One was RNA sequencing. We just went for broke, and let's look at everything. And what we found, we were actually surprised at the number of genes that were impacted. And you can see from the volcano plot here, there's over 2,000 genes that were upregulated and downregulated with the treatment, AGE treatment, which we were very surprised by that. And we did some pathway analysis, and I'll point out a few key ones here. Diabetes was a significant pathway affected. Cell survival and viability, cell proliferation, and even connective tissue hit as a target. So wow, we're like, this is perfect. And even when we look at specifically at ECM genes, over almost 50% of those genes were impacted. So this was very exciting for us. And we did some other experiments to be more targeted rather than just, the shotgun approach with sequencing. And we did some standard cell culture assays. We monitored cell proliferation with EDU staining, which monitors active replication in the cells. We did some targeted PCR, which I'll show you a couple examples of. And we did some mitochondrial respiration assays using the Seahorse platform. And you can see graphically here, hopefully, oops, if I don't crash into the computer, here's our 0 AGEs and now down, and what you see is the pink is the proliferating cells, and you see less pink as we move down. So if we plot that, we see that AGEs dose-dependently suppress cell proliferation. We measured several cell proliferation genes using ddPCR, MYBL2 being probably one of the more affected ones. And we see, again, a dose-dependent suppression of the cell proliferation-related genes. Now, keep your mind on that one because we'll come back to that in a little bit as well. And interestingly, when we ran on the Seahorse platform, which one of the pieces of data you get out of that is ATP production. And we found a dose-dependent reduction in ATP production, even with increased mitochondrial biogenesis or increased mitochondrial content that we assessed as well. So the cells are trying to compensate for it, but just there's something going on that won't let them do what they need to do. And so then our next step was, okay, if we're seeing this in cells, can this translate to animals or humans? That was our next thought. We want to take it to humans eventually. So we did a couple assays to start that process using cell culture. So I know I just said I was translating, but I'll get to that in a second. We utilized first a traditional wound assay, which is super simple assay. You fill your cell culture dish full of cells. You take a pipette tip, scratch it down the middle, and watch the cells migrate and fill it back in. And if they don't do that as well, it's an indication that maybe if it was an actual true injury, they wouldn't heal as well. And we saw what we hypothesized we would see if you compare 0 AGEs to 200 micrograms per mil. There is some gap closure compared to control, but it's nowhere near the 0. So now that gave us some thought, okay, there's potential translation to wound healing. So the next step was, okay, before we do our first mouse experiment, do we think we would see this in a mouse or another animal? So what we did is we had, this was one of those freezer saves that turned out to be a good thing for me. We had saved some blood from rats with streptozotocin-induced diabetes. So it's a model of type 1 diabetes. The animals have elevated AGEs and elevated blood glucose levels. So we took our cells and then treated them with serum from these rats with and without diabetes. And this is the same assay. This is the wound assay, but here I have it laid out as percent gap closure. And we have our uncontrolled diabetes on the bottom, and then our ones without diabetes, and then a control experiment. And you can see that the control and the non-diabetic serum, the gap starts to recover where we get no recovery in the diabetic sample. So suggesting now with blood from these animals, we're seeing what we want to see. Don't ask me about the 9-hour, ask my student who can't tell time. But they're supposed to be, obviously you can do the math. So in summary so far, and then we're going to take a maybe third or fourth inning stretch, and then we'll move on here to some other stuff. We know that AGs dramatically limit tenocyte proliferation. We see impairments in cellular respiration and mitochondrial function. These observations appear to be largely independent of glucose levels, at least in our cell culture model. And we see cell proliferation and migration inhibition in this model as well. So that then was kind of where we thought, okay, we need to start doing something in an animal model. Okay, and we have 3 approaches that we're doing. 2 of them funded through the R1 and 1 that we're trying to get funded for another R1, but we're doing some of the experiments now anyway. And 1 model is a DBDB model. We're doing some work in that, which I'm not going to show that today. And we're currently developing a Cree a mouse that will have a tendon-specific rage knockdown. That will be available hopefully in May or June, and we'll start some experiments on that. We're halfway through the breeding process. What I'd like to show you today is some work we've done in a normal, healthy mouse. We started with this model because we wanted to be able to just isolate the effects of AGEs and not deal with all the other things going on with diabetes. So we designed an approach where we use the DBDB heterozygous controls that we get for free when we buy our DBDB mice, so we might as well use them for something. And we treat them with either a vehicle or a Zaleron, which is a rage inhibitor, rage antagonist. And we decided to go with 3 weeks of treatment for our initial experiments because we wanted to focus on mechanical changes, and 3 to 5 weeks is for tendon is a reasonable time to do that. And so what we did is we exposed the tendon. You can see the patellar tendon right here, and we essentially poke a hole in it. Okay, we have a little biopsy needle, and we go and take a chunk out. And then we sew them back up, and then they get to simulate the cell culture work. We inject them with either BSA or that AGE BSA that we created and we use in our cell culture, exact same molecule. And you can see what the hole looks like here. So it's a pretty good size chunk, not quite a full central third remover, but it definitely impacts their mechanical function. And we've started analyzing this, and I'll show you some of the initial results. If you look at tendon stiffness, so I guess it's a step back. Once the animal's euthanized, we pull the tendon out. We do tensile testing on it, basically clamp the tendon down on 2 ends, and you pull it till it breaks, and you get failure stress and various other mechanical properties like stiffness and such. And if we look at the stiffness, we see some kind of interesting results here. It's pretty clear that the AGE treatment, which is the black bar, is suppressing the healing of the tendon. All 4 of these bars are injured mice. So we would want to see if the AGEs had no effect, this black bar would be the same as the Purdue gold, non-sanctioned Purdue gold color that I chose. Don't tell Purdue. But when we look at tendon modulus, which is derived from the stress strain curve, so we measure the stress levels as we're pulling on it, and we measure the strain levels, and the slope of that curve, I'll show you some later, is modulus. And stiffness is impacted by the size and internal structure of the tendon, the cross-section area tendon, whereas modulus is primarily a reflection of the internal structure of the tendon. So when we measure modulus, we see a large reduction with the AGEs, and when we give them the rage inhibitor, it largely rescues the effect of our exogenous AGEs. So very exciting because that's exactly what we hypothesized. The stiffness one is a little strange. I'm not going to delve into that because we're still trying to figure some of that out, but I think it might have to due to some extent to potential changes in the cross-sectional area of the tendon because stiffness is affected by that. And when you operate on the tendon, it swells up by about 30% due to scar formation stuff. But it's not the stout normal structure. Interestingly, though, even though the modulus went down a lot, we don't see a difference in the failure stress or failure load of the tendon with either AGEs or the rage inhibitor. But we do see some differences in strain. But overall, not bad start. We might try some different doses. But in general, I think the graph on the right is really what we were hoping to see. And we've done some follow-up PCR just to link this back to the cell culture. These are a lot smaller group sizes. We're actually running those experiments right now to get the data for PCR, because once you put the tendon through the biomechanical testing, it's no longer useful for PCR. So basically have to run everything twice. And referencing back to the MYBL2, that was that one gene that was suppressed a lot in our cell culture model. And we see the same thing here when we give the cells, or excuse me, give the mice, AGEs, it suppresses MYBL2. And when we give them the RAGE inhibitor in the presence of AGEs, it rescues that response. And we see similar effects with, this is a mitochondrial related complex 3 gene, similar suppression and renewal with the RAGE inhibitor. And we also see an impact on the RAGE receptor as well. Again, very preliminary data, small n size, but so far it's largely showing what we saw in cell culture and translating it to a mouse model. All right, I'm going to take a deep breath. And now we're going to get into the fun more more exciting area, because I'm trained as a human guy. But of course, as was mentioned at the start, we kind of go from the cell culture all the way to the humans. So this is the other half of the R01 that we're working on. We got an initial pilot study to do. We studied around 40 individuals. And now we're pushing that. Hopefully by the end of the 3rd or 4th year, the grant will have us up to around 160 people, so much larger cohort measurements. So I'm going to tell you a little bit about some of the methodologies we use and what we're getting with this data. Our goal here is to tie AGEs to tendons in humans, is what we're trying to do. So we started out with an age-matched cohort here. And you'll, as you would expect with the populations, the patients with diabetes had higher A1Cs, fairly well controlled. I mean, we had a couple individuals out in the 7s and 8s, but generally leaning around 7 for A1C. And what we did is we brought them in, we took a blood sample, and we did 2 primary tests on them. One is our ultrasound assessment, which I'll walk you through here. This is obviously me, maybe. I don't know why I cut my own head off. I guess I just don't want you to see me. But what we have is a setup where we affix an ultrasound probe over the patellar tendon. And as a running joke, we need a long probe, long linear probe. The only one we could find is a vet rectal probe. So whole brand new, and no, we don't study rectal function, but we strap them in here to a leg strap, and this is hooked to a load cell and to the ultrasound. We have them perform an isometric contraction, and we collect a video image of the tendon during the contraction. What you end up getting out of it is the tendon actually goes from a slack state to a unslacked state, and we can measure that with a friend of mine at BME that has a strain tracking software. We essentially draw ROIs around the tendon, play the video, and it measures the strain in the tendon during that contraction. We have to do a 10-second contraction. So we get strain data from that. The stress data comes from the load cell measures force output. We use, we get the tendon cross-sectional area. Stress is force over cross-sectional area. We get the cross-sectional area data, like I'll show you in a second, from MRI imaging, which is the second assessment we do. And then the slope of this curve is the modulus or stiffness of the tendon. So we can measure that in vivo in these individuals. For the MRI, what we do is we develop this approach we had to bug Siemens to get a unpublished WIP that they have, the unpublished sequence that they don't use clinically. And basically what we do is we set the individual up to assess the patellar tendon, which you can see here, and we run a special ultra short T2 star sequence that collects multiple TEs. And through a bunch of math that I'm not going to get into, we can come to a measurement where we can get the signal that we end up getting from this assessment reflects the actual organization of the tendon. Okay, so the way the tendon is set up, if the I guess we don't have time to get into details, but it works out really well. You do this special scan, and that the output you get from that scan reflects the organization of the tendon. And other groups have shown that this signal here measures T2 star signal correlates really well with things like tendon modulus This V-Zip-P is a clinical survey of tendon function. The higher the score, the better. And then what we also can do to measure the morphology and the size of the tendon, we take hundreds of slices, or not hundred, well, close to a couple hundred slices down the length of the tendon, and we can then 3D reconstruct the tendon itself and measure the size and volume of the tendon with this MRI. So we get those, and what did we see? Well, floating back, remember I mentioned that we thought tendon should get stiffer, and we saw the other groups have seen with the Achilles that it does not. Well, we're seeing the same thing in our patellar tendon in our humans. This is our controls, our patients with prediabetes and our patients with type 2 diabetes, and we see the progressive reduction in tendon modulus. And we were really surprised by this because these individuals, a lot of them didn't even know that they had prediabetes. We were screening them for the control group, and they had a higher A1Cs. So it seems to be kicking in early on. And then when we look at the size of the tendon, so normally you'd see like exercise training, building muscle mass, that normally increases the size of your tendon a little bit. So we thought with these individuals, with many of them being obese or overweight, we might see a larger tendon because of that chronic loading. But in fact, the tendon is smaller in our individuals with diabetes. So we're seeing some of those things. Now, what's the next step here? How do we link in AGEs and stuff? Well, I mentioned a blood draw. And so what we're doing now, and this is really one of the key focuses of the grant, this is our first 18 subjects, is we're measuring that various AGEs. We're measuring all 5 different AGEs in their blood, and we're trying to correlate that with these tendon properties that we're measuring, like modulus and here tendon volume. And we're actually starting to see some pretty exciting results in that the more CML you have, the lower your modulus. The relationship's a little bit stronger when you just include the red dots, which is our patients with type 2 diabetes. Again, very small sample size yet, but it's looking promising. The volume data is a stronger correlation, showing that the more CML we have, the greater the tendon volume. And higher tendon volume can be a good thing, for example, with exercise training and things like that, but it's also a sign of tendon pathology. Typically, those with that pathology have larger tendons. And if we look at that clinical V-Zip-P outcome, again, the higher the score, the better off the individual is, and that higher score is associated with lower CML levels. And so everything is kind of going the way we hoped. And one last thing that I wanted to point out is we're not just looking at AGs, because we all know with diabetes, there's a hypercholesterolemia, things like that. So we actually screened about 40 different serum variables, and we have a collaborator who's a very, very smart statistician, and he applied this what's called elastic net to all our data. And what it does is it screens for interactions between various covariants. It tries to work through all the covariant noise to pick out the covariants that are most likely involved. And what was the most interesting finding was that we can increase our predictive potential utilizing more than just one variable. We haven't mixed in AGEs into this yet. We just don't have enough N size. But you can see that, for example, tendon cross-sectional area, if we include these variables here, we can predict about 70% of the variation in tendon volume. And what was really interesting about it is, even though I just showed you that when I grouped the patients by those the prediabetes and diabetes, when you do regression analysis and look just as group as a predictor, it explains actually very little of the variation in tendon properties. It's actually this combination of all these other variables that are involved. So we plan to take that much further as we go along here. I'm going to skip over this since I kind of said all that. So I know I'm going to take a little bit longer than I planned. So I think I have time for maybe just one more of these topics. And so I guess since this one's queued up, unless you want me to talk about the estrogen topic. Protein. All right. So I appreciate you being patient with me and letting me show some of these different things. We are doing, as I mentioned, some work with estrogen. We got an R1 scored in that area, and we're really interested in how tendon health changes across the lifespan in women, and how exogenous estrogen and maybe other hormonal changes impact those changes. And what we're trying to do is basically chart just loss of muscle function and mass, loss of bone, and tendon. Do those all correlate with each other, or do they have different trajectories throughout life? And we got a solid first score, and we're coming back for a second round. And now I'm going to talk about this a little bit because it leads to that health of the forces things. And since this is a nutrition group, basically where this idea came from was, well, one, my background was in protein synthesis and isotope tracers when I was younger. And I think many of you know Lane Campbell from Purdue is a prominent researcher there in nutrition. He approached me one day with a call from the beef checkoff about muscle stuff. And I'm like, well, we should do a tendon project. And he's like, what? Tendon to protein? So I showed him some literature. And long story short, there's some data in rodent models that show that specific amino acids, leucine, glycine, maybe hydroxyproline, seem to have beneficial effects on recovering tendon injuries. And there's a few human projects that show that people recover better from tendinopathy when they're given certain protein, like leucine or certain amino acids or high protein. And then about 10 years ago, a group came out in young adults showing that if you supplemented individuals with whey protein, which is the black bar, versus no whey protein, and ran them through a chronic resistance training study, you had more tendon hypertrophy. And I'm like, wow, that's great. That I wasn't expecting that. And one of the issues that we've noticed in older adults through research I did as a postdoc and a couple other groups is that in young adults, when you do train them, they do get tendon hypertrophy. Older adults don't seem to have that adaptation. Their tendons seem to be more resistant. So Wayne and I were like, well, maybe we can use protein or beef to advance that. So the first experiment we did, because the first question we got asked is, well, there's no way there's amino acids getting to the tendon because the tendon's avascular and stuff like that. So we had to overcome that hurdle in the review. So one of the techniques that we've used in my lab is microdialysis, and I'll explain how that works here briefly. But essentially, we insert a microdialysis catheter right next to the Achilles tendon, so we get everything that's bathing that tendon, and we then flow saline-based solution through it. And if you're familiar with your cardiovascular physiology, it works just like a capillary, osmotic, oncotic pressures and hydrostatic pressures, and stuff moves back and forth through the holes in this microdialysis catheter. And when you push your fluid through, as it goes through, stuff comes in, stuff goes out. But since there's nothing in it, it's only stuff coming in, and we get our molecule on the other side. So we recruited some young and older adults, gave them an amino acid bolus, a mixed amino acid bolus, put the catheter in, and measured the changes in amino acid concentrations at the level of the Achilles tendon. And lo and behold, we saw a nice, robust increase in amino acid levels. So we solved that problem, and I'm not sure if that's why the grant got funded, but it was a cool study. So long story short, you might ask, well, why do we care about the beef side? Well, interesting with beef is consumption of it, you end up with higher glycine load. So that was one sell that we made to the reviewers. And we also came up with, a higher protein scheme. We chose 1.4 grams per kilo per day because that's roughly what 95%, roughly what 5% of older adults achieve is like kind of on the high end. So our goal was 1.6, but we wanted to be a little bit more realistic. And this was protein spread throughout the day. So we didn't give them like a big drink or anything like that. And then we ran them through a 12-week resistance training program, focusing on the lower legs. And we focused only on adult women. One, because they're more vulnerable to sarcopenia, but also that was what the company was more interested in. And we chose a protocol, that exact same protocol that had been used in young adults to induce tendon hypertrophy. So we had a model that we think would work. And just to kind of highlight this is our group characteristics here briefly. And as I mentioned about the higher leucine glycine, if you look at the beef group compared to the normal protein group and even the high protein mixed protein group, they had higher leucine and glycine in their blood throughout at least at the 3 points that we measured it. This is their actual protein intakes. So we did hit pretty close to our 1.4 grams per kilo. So to make this really fast, I'll summarize this in one sentence. This was not my title that I chose, but this is what we had to put. And surprisingly, we didn't really see that much happen with the high protein group. So a little bit disappointing from that standpoint. However, if you look more closely, and me being a tendon person, if you dive a little bit more into the protein, I think the data is a little, even though it wasn't quite significant because of the lower end size, keep in mind that no study that I know of, that I can at least think of off the top of my head, has shown tendon hypertrophy in older adults in 60 to 80-year-old range. But when you look at the actual regional changes in the data, we actually saw some growth of the tendon. And I know it doesn't look like a lot, this is percent change. It doesn't look like a lot, but in a young adult, the most you get out of 12-week protocol is maybe 5 to 8% max in tendon hypertrophy. I mean, you see that much in muscle as well. So we're hitting like 6 or 7% here in overall tendon size. And we didn't really raise amino acid levels to the extent that we might have if we had given like a bolus during training and things like that. So I really think that there's some more work we can do here. And in fact, that's essentially what we're doing. We received a pilot grant. This is my student, Eric Gutierrez. He's up for promotion to Lieutenant Colonel here shortly. 15 years in the Army, and now he decided to get a PhD. So the Army is allowing him 3 years to get a PhD, which is really difficult because if we don't get him done in 3 years, him and I are basically in big trouble. So we have a very aggressive study plan. And we reached out to Usarium, which is an Army Research Center in Natick, and found out that they were actually doing some collagen protein peptide studies with muscle. And when we mentioned tendon, they're like, whoa, that's awesome. And they wanted to do a healing model. So we got a pilot study where we're actually cutting a hole in the Achilles. We're giving these mice a protein peptide compound for 2 to 5 weeks. And we're going to measure the healing in the tissue with MRI, 7T MRI and our functional properties. And then hopefully the requirement of the pilot funding is to submit a full DoD grant. So hopefully that will lead to a DoD grant. So with that, I'm just going to do the last little thing here. I got to thank everybody. The one mantra I have in my lab is there's no I. Nobody says I about everything. It's we on everything. And we have a great group of people. One of my favorite things is that once I find a great group of people, I don't want to let them go. But this is our lab group here at Purdue. Jay Haas in Michigan did all the AGE work. Talia here is a PhD student in Nat Diamond's lab at University of Pennsylvania. They've helped us with a lot of the mechanical work. Our 2 statisticians that have been great, and our BME collaborator, Craig Gorgian. And then all the cell culture work was done by Shivam, who is now in his first year of orthopedic surgery residency. And then all of our funding sources that mentioned there. So thank you very much. I appreciate it.
Well, I have a question about the source of AGEs. So you were using bovine serum albumin in mice. Why not to use rodent source?
That's a good question. I think the honest thing, I probably didn't think about it at the time.
Can you buy it? I looked. Can you buy mouse BSA?
Well, you can isolate it, I believe.
Yeah,
Not necessarily mouse, the rat would be
I
fine.
tried to think about that, and you'd have to kill a lot of mice.
Yeah, that's a good point. Yeah.
No, I looked for it, I'm not sure, and so we can't call it BSA,
Yeah.
because that would be bovine serum, and we just.
Yeah, I think-
anything from mice.
Yeah, some of it probably was stemmed from that protocol to make those AGs was pretty well established in the literature. And we just went with that approach.
Also, I made that,
Okay.
we put BSA in primates, and it doesn't cross-react, it's
Yeah.
like 99.9999% the same.
Also, I noticed that you used glyoxale to make AGE protein, right?
Include how that driven, yeah,
Not glucose.
yeah, correct.
Yeah, because it's believed that the major age comes from glucose rather than from any other short sugars, which is called glucosipane or something.
Yeah, I know what you're talking.
I saw that
Yeah.
you made picture. So it might be more relevant for future studies, and
Sure.
especially if you connect this to markers of, well, let's say, inflammation,
Yes.
because
Yeah, that's that's what we're trying to do with the serum analysis. We've looked at like CRP and some other inflammatory markers, and I think maybe when we get a larger number of individuals, we might start seeing a more refined number of things. We did assess, I didn't show, we did IL and beta, we've done IL-6, we've done TNF alpha, and none of those lit up like leptin or leptin was the big individually as a single predictor, doing just like a Pearson, it's like it was the strongest predictor. We just haven't had time to follow up on.
Free fatty acids, because those will be up with.
Okay.
Can you go up to, you had a correlation, and you had all of the groups were going the same, I think it exemplified, we had all the group there, so this is what you said, that the relationship is not different by group.
Yeah,
All those, yeah.
yeah, no, thank you for bringing that out. I kind of got in a hurry and I forgot to say it. That is the key point, key takeaway is that if you look at just patients with diabetes, sometimes the correlation is a little stronger, but when you look at everyone as a whole, it's not whether you're a patient or a control, it's whether your CML is high or low, or your the same thing with yeah, exactly. That's what, yeah, I know. So it's not whether it's whether you're a patient, it's whether those are high or low. So if you're a patient with diabetes, theoretically, what this would mean is if you're a patient that can reduce your AGE load either through dietary modifications or maybe exercise, you might make your tendon better, even if your blood sugar isn't quite perfect.
Yeah, and so if it's not, the AGEs you're saying are not exactly tied directly to blood glucose. There's something else.
I mean, they theoretically are, but there's so many other sources of AGEs that are either coming from the diet or from the glycolysis that's not allowing for that relationship of glucose to be there like we would expect.
Questions now?
So do you see any difference based on, like in the AGEs, based on diabetes and severity? So for example, in individuals that have neuropathy compared to those that don't.
Yeah, I think other people have shown it. We haven't gotten a big enough number of individuals to really tease that out yet. And we also haven't had very many patients with poorly controlled diabetes. That was the one interesting, or not one interesting, I don't know why I say that, but an interesting thing from the study is as we were recruiting individuals, and I ended up noting this in the limitation of the paper, is that, pretty much any individual with diabetes that has access to healthcare is on some sort of medication. So we found that that's one hiccup with this. We don't have enough individuals to start throwing in medications into the equation, the regression models. Would I get, if I had to guess, they're probably higher, but that would be an educated guess.
Other questions about anything? Yeah.
We did,
Use beef source if you're suggesting that age. Cooking beef.
Oh, yeah.
Be a high source of.
Yeah.
Age of blood.
Wayne and I have talked about that a few different times, we just haven't gotten to it yet, we do have saves. And, that's been a point of discussion of, measuring AGEs in those individuals. We actually have another paper we're working on too, that we measured a whole bunch of cardiometabolic risk factors in those, individuals. There were some improvements in the, both in the high protein groups on a kind of separate note, that'll, whenever we get around to writing it. But yeah, that definitely, I definitely agree. That would be really cool.
There's another one there, yeah.
This may just be due to my lack of really ever thinking about attendance, but so you were surprised when you saw like a decrease in stiffness in the diabetic groups in mice. At what point is like, there's gotta be a curve, right, where like a certain amount of stiffness is good, but a certain amount of like a lack of a certain amount of
100%.
flexibility is like poor for
You
stability.
just got an A in attendance. Absolutely. There is a sweet zone where you want. I won't pull the slide. I can just say it, but there, if you look at stiffness within normal healthy ranges, higher stiffness is correlated with more muscle power output, a better running economy. Like if you look at, for example, if you look at a sprinter, they almost always have much stiffer tendons than a non-sprinter. Thus, and so they're stiff, but they're brittle. So you're exactly right. There is a sweet spot for it.
So do you think those diabetic mice might be going on the other side of that curve, like towards a little?
Are so unhealthy that they become so unstable, lax or whatever. I can't think of the right word. That's probably significantly contributing to functional problems, both in humans. So where, I mean, if you look at the animal data, the changes in stiffness are actually on par with the human differences that are seen too.
Since this was good news, Thursday, and our department didn't have any good news, I'm going to add a piece of good news about our speaker. And that is his leadership has been noticed at the national level, and he's just been chosen to be a participant in the American Kinesiology Association's Leadership Institute, which is a year of a lot of work. But congratulations on that, and thank you very much for this.
Darius